Assalamualaikum. I am Nur Ayuni binti Muhammad Nur. I'm going to explain about steps of DNA extraction. Steps of DNA extraction. Step 1. Pipette 1.5 ml of E. coli culture into microcentrifuge tube in the laminar flow. The airflow was switched on to ensure no contamination occur. E. coli culture tube was placed in opposite direction in centrifuge machine and was centrifuged for 1 minute at speed 12,000 rpm. After centrifugation, 2 layers of culture was produced, which are supernatant at the top and pellet that contain the cell at the bottom. Centrifugation is the process that involves the use of centrifugal force for the sedimentation of the mixture. Centrifugation alters the effective gravitational force on tubes so as more rapidly and completely cause the precipitate, or we call as pellet, to gather at the bottom of the tube. The remaining solution is properly called as supernatant. The supernatant liquid is quickly descended from the tube without disturbing the precipitate.
Hi, my name is Dini Shakira and I'll be explaining on the procedure from step 4 until step 6. On step 4, the supernatant that we acquired before this is poured out into a beaker and then the tube is left to dry for a while. In step 5, 100 microliters of lysis buffer is added into the tube, and then the tube is resuspended by vortexing at high forces. Lysis buffer is used in step 5 because lysis will destroy the fatty membranes that envelops the cells, as well the nucleus membranes within the cells. This will result in the breaking open the cells that will release the DNA. In the step 6, 20 microliter of proteinase K is pipetted into the tube, and after that, the tube is incubated in the water bath for 15 minutes long at 55 degrees Celsius. The functions of proteinase K that was used in step 6 is to break down many contaminating proteins that were present during the DNA extraction process. Other than that, it's also to provide protection to the nucleic acid from the nucleases attack by degrading the nucleases that may be present in the DNA extraction process. Proteinase K is also catalyzes the protein hydrolysis. Put in mind that the tube must be flicked every 5 minutes to ensure the proper mixing.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Alia binti Abdul Nasir. Next, I will explain the next following step. Step number 7. After that, 20 microliter of RNase A was added to the cell, vortexed to mix it, and left at room temperature for 2 minutes. RNase A was utilized in this step since it is an enteroribonuclease that hydrolyzes pyrimidine residues in RNA. It also breaks the phosphodiester connections to another nucleotides. How did RNase A work in this step? So basically, it will break the phosphodiester connection to another nucleotide. It does this by cleaving all forms of cellular RNA that are not essential for cells. It is particularly useful for cleaving single-stranded cellular RNAs. RNase A was used because it can eliminate RNA during plasmic and genomic DNA separation procedure. Step number 8. In this step, binding buffer was added in the amount of 500 microliter and transferred to a spin column. The spin column was then centrifuged at 1200 grams for 20 seconds. What is binding buffer? First and foremost, binding buffer was intended for use in the DNA clean and concentrated product line. In this phase, the binding buffer serves the following purposes. This buffer enables effective DNA binding and recovery from enzymatic processes and impure DNA samples. The highest DNA binding of any column technique is achieved with the cryotropic salt binding buffer. Wash buffers that are strong enough eliminate all traces of protein and salt. It also allows for pH stability of DNA in storage. DNA is eluted in a low salt buffer. Step number 9, the spin column's collecting tube will be removed and the flow-through will be discarded. That's all for my part.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Kaisha Amira binti Zaisam. I will explain step of DNA extraction from 10 to 12. So step 10 is 500 microliter clean buffer was added and the spin column was centrifuged for 20 seconds at 12,000 G, and then the flow through was discarded. An ethanol solution was used in cleaning step to wash the nucleic acid. It is to increase the purity while keeping DNA bound to the silica membrane column. These washes remove contaminating proteins, lipopolysaccharide, and small RNA. Step 11 is 500 microliter of wash buffer was added and the spin column was centrifuged for 20 seconds at 12,000 G, and after that the flow through was discarded. Wash buffer is used in this step. Wash buffer are alcohol-based solution for removing proteins, salt, and other contaminants from sample and upstream binding buffer. Other than that, a wash buffer is used to rinse tissue sections, effectively removing the previous reagent and preparing the tissue for the application for the next reagent. Step 12 is the empty column was spun for 1 minute. The DNA at spin column was eluted into microcentrifuge tube and stored it at -20 Celsius. Next, I will explain the elution step. Elution of DNA is the process of extracting DNA from material by washing them with a solvent, typically a DNA elution buffer. Elution buffer is used to break up association between a protein that have previously been separated and the chemical that bind it. Lastly, I will explain the difference between spin column and collection tube. Spin column have membrane silica gel to filter DNA, while collection tube does not have it. Besides, spin column function in binding nucleic acid to its membrane, while collection tube accumulate other contaminants like buffer. That's all from us. Thank you.